Hello, I am Hugo Talibart, postdoc at the National Museum of Natural History in Paris, and this poster outlines the work that was mainly carried out during my PhD at INRIA Rennes with François Coste, and recently published in BMC Bioinformatics with the title PPalign Optimal Alignments of POT Models Representing Proteins with Direct Coupling Information. Thanks to sequencing technologies, there's an increasing number of protein sequences available, and in vivo and in vitro annotation methods cannot keep up with this pace. To annotate them in silico, the main approaches rely on significant alignments of carry sequences to annotated proteins or protein families using BLAST or the current state-of-the-art methods based on profile hidden Markov models. These methods are powerful, but they do not take coevolution between residues into account, and therefore they discard valuable information on higher order constraints, such as structural contexts. We propose to take advantage of recent advances in the field of contact prediction and to represent proteins by POT models, which model both positional composition and direct couplings between positions, and to compare proteins by aligning these models. In the context of proteins, POT models were originally introduced by direct coupling analysis for contact prediction. A POT model typically represents a multiple sequence alignment of a sequence of interests and its close homologs. It can be defined as the statistical model which maximizes channel entropy and generates the empirical single and double frequencies of the letters in the multiple sequence alignments as its marginals. Its probability distribution has the following form. The probability of a sequence which is the same length of the multiple sequence alignments is a normalization constant times the exponential of a sum of parameters evaluated at each letter of the sequence. POT models have two types of parameters, field vectors and coupling matrices, which can be assigned a practical interpretation. Each field vector Vi represents the composition of column I in the multiple sequence alignment, where Vi of A reflects the propensity of letter A to be found at position I, and each coupling matrix Wij represents the compatibility of letters A and B to be found together to co-occur at positions I and J. These models were successfully used for contact prediction, since the norms of these pairwise coupling parameters are great indicators of a direct interaction strength between two positions. We believe that in addition to their application to contact prediction, protein-protein interaction prediction, and mutation fitness landscapes, POT models represent an interesting alternative to profile hidden Markov models for sequence comparison, since they can model both positional conservation and pairwise dependencies reflecting coevolution constraints, and we propose to investigate on their performances in homology detection. As a first step, we introduced PPalign, a pairwise POT model alignment method, which can provide in tractable time an alignment of two protein sequences by aligning the two POT models representing them. We relied on an existing solver initially dedicated to protein structure alignments to formulate this problem as an integer linear programming problem, where under constraints ensuring that the alignment is proper, a similarity score between two POT models is maximized. We base this similarity score on the scalar product. Thanks to this efficient solver, a solution within a chosen epsilon range of the exact solution can be found in tractable time, 1 minute and 37 seconds on average in our experiments. Its performances in terms of alignment quality were assessed on a benchmark which we built by extracting a non-redundant set of low sequence identity pairwise sequence alignments from the database of manually curated structural alignments CFUS. In these experiments, our alignments of post models had slightly better F1 scores than the alignments of profile hidden Markov models by HH-align, and even better when the sequence identities were the lowest. We were mostly outperformed when alignments had more gaps. We showed that direct coupling information could improve the quality of sequence alignments, in some cases significantly, which suggests that post models might be a valuable alternative to provide the Markov models to perform alignment-based homology search. In some cases, however, our alignments were worse when taking this coupling information into account, and we are currently working on improving these results. Post models were initially designed for direct interaction prediction, and we are now focusing on how to build post models better suited for pairwise comparison, in particular by making them less sensitive to small sampling variations. Thank you. I would be happy to answer your questions.